Everyone. So this would be chapter 1 of our analytical chemistry subject, and specifically we have lesson 1.1 here, which will talk about the nature of analytical chemistry. So what is analytical chemistry? So analytical chemistry is actually considered a measurement science since it deals with the quantitative and qualitative aspect or determination of the substance present in a certain compound or a certain or given sample. So it actually is composed of a set of very powerful ideas and methods which had been developed and eventually improved throughout the years. It's mainly a very useful field of science as it could be applied to almost all other fields of sciences, including your field of specialization, which would be the environmental science. For the role of analytical chemistry is actually regarded as a central field of science, as you may see here in this diagram, since almost all ideas or methods of analytical chemistry can be applied to, say, chemistry or to other areas of chemistry, including biochemistry, inorganic chemistry, organic chemistry, and physical chemistry. It could also be applied to physics, which we will show later on, or in material science. And even in engineering, it could be applied, as well as in medicine. Also, some social sciences could also use the methods that could be learned when you try to study analytical chemistry. Aside from that, we could also have biology, geology, and again, as what I have mentioned a while ago, the environmental science. For agricultural students, analytical chemistry is also very much useful, especially when you do crop science and food science, as well as soil science. So for the role of analytical chemistry, as applied to environmental science, it could be used to quantify hydrocarbons, nitrogen oxides, and carbon monoxide, as well as carbon dioxide in air. So you could actually do an environmental monitoring on the quality of air around you, as well as for water sources, sediments, and dump sites. So you could actually assess the level of pollution with regards to the presence of heavy metals when you try to learn some of the methods in analytical chemistry and apply it later on to environmental science. The next one, it could also be applied to industry. So we have in the food industry, you could actually determine the nutrition content of food products. So the techniques that you may learn in analytical chemistry could also be applied in this aspect, as well as the elemental analysis in steel carbon. So you could analyze any industrial products for their elemental composition, and again, analytical chemistry or your learnings in analytical chemistry would be the key factor here. Aside from that, it could also be applied into medicine. So some of the examples, as you may see here, could be in terms of blood samples. So it could, your knowledge in analytical chemistry will help you to apply it later on in medicine, such as determining the concentrations of oxygen and carbon dioxide in blood, also the ionized calcium present in blood serum, or even testing your blood sugar levels using a blood glucose test. So it actually applies the concepts learned in analytical chemistry. As we have also discussed a while ago, another role, and this one is quite important, as analytical chemistry or knowledge in analytical chemistry has been applied also in the exploration of the planet Mars using the Pathfinder mission. And it actually deploys the Sojourner, Spirit, and Opportunity rovers. And with that, examples of which that had been applied into that exploration of Mars include information on chemical composition of rocks and soils present in the Martian planet, and usually, or is actually employ the use of alpha particle X-ray spectrometer. Also, the Mars Science Laboratory aboard the Curiosity rover contained an analytical chemistry lab. So it was actually used in the determination of different elements present in the Martian atmosphere as well as in the surface of the planet. So there were surveys conducted on the carbon compound sources for the planet as well as the organic compounds that could be important to life. And with that, it was also used whether or not life could be present, or to conclude that life could or could not be present in that planet. And also, lastly, we have also the determination of the composition of the Martian atmosphere. So with that, let's proceed with the concept of quantitative and qualitative analysis, which are the main concepts that we need to learn first when we study analytical chemistry. So let's try to differentiate the two. We have qualitative analysis as the first one. So when you do a chemical analysis, this would be the first step, which would be the establishing of the chemical identity of the species inside a sample or contained in a sample. So the first thing you need to do when you conduct a chemical analysis or analytical chemistry application would be the conduct of qualitative analysis. So the next one would be the quantitative analysis. So this one now determines the relative amounts of the chemical species in numerical terms. This means that as you have already established the chemical species or identity of the chemical species in the sample, then you could quantify or determine the amount of those chemical species and report them in terms of numerical terms. So, say a concentration or the mass, or it could be the volume of that particular chemical species in the sample. So that would be the quantitative analysis. And of course, when you do chemical analysis, both are actually required. So you need to determine first the qualitative information of your sample, and after that, after determining that qualitative information, then you can proceed to the quantitative determination of those chemical species. And when we talk about quantitative analysis, we should know the different quantitative analytical methods. So when I say quantitative analytical methods, these are the methods that could be applied for us to quantify the relative amounts of those chemical species being identified. So the results of a typical quantitative analysis usually comes from two measurements. One would be mass or the volume of the sample, or it could be the quantity that is proportional to the amount of analyte, be it a mass, volume, intensity of light, or electrical charge present in the analyte or sample, which is proportional to the chemical species that you are referring to. And it should be in a final, or this should be the final measurement. So for the quantitative analytical methods, the first one would be the gravimetric methods. And the focus of gravimetric methods is the mass of the analyte or the mass of compound chemically related to the analyte. Example would be the copper coated on an electrode. So you could actually quantify that one in terms of the mass. Or it could be the amount of chloride ion or chlorine precipitated as silver chloride in the sample. So if you have no means of quantifying the chlorine present or the chloride ion present in a particular sample, then you could do reactions which will result to the precipitation of that particular ion into a certain ionic compound or an insoluble ionic compound. So example of this one would again be this chloride or chlorine precipitated as silver chloride for us to quantify the amount of chlorine or chloride ion present in the sample. The next one would be the volumetric methods. So from the name itself, you could derive that it focuses on the volume of a solution with reagent that reacts completely with the analyte. So again, this is not directly the amount of analyte present, but this is with regards to the amount of the reagent that reacts completely with the analyte. So examples would be, or an example would be, the most common one that we'll be using would be via titration methods. And the chemical species that is, or the reagent that is mostly used would be sodium hydroxide. And with that, we could determine the amount of, say, HCl or hydrochloric acid in a sample by reacting it with a certain volume of sodium hydroxide at a known concentration. And with that, the important thing also that we need to establish would be the balanced chemical reaction between the two reagents or between the two chemical species. Aside from volumetric methods, we also have electroanalytical methods, and this would actually be using the electrical properties of a solution. It could be the potential, electrical potential, current, resistance, or the quantity of electrical charge. More of this as we proceed on to our later chapters. And then we also have spectroscopic methods. For the spectroscopic methods, we will be dealing or exploring first the infrared or IR and the UVVs or ultraviolet visible spectroscopy. So the main concept for this one is that the analyte could have an interaction with the electromagnetic radiation that is beamed towards the analyte or solution. So it could either be an absorption or via a luminescence, and there are spectroscopic methods available for that. Or it could also be the production of radiation by the analyte. So some analytes could actually release a certain form of radiation which could be later on measured. Or it could be via the emission spectroscopy. And then with those, we could also have some other miscellaneous methods. When we talk about the mass to charge ratio or the mass with respect to the charge of the analyte present or an ion present. So we could employ the concepts or methods of mass spectroscopy, but we will not be discussing these miscellaneous methods. We will focus mainly on the electroanalytical methods and the spectroscopic methods, aside from the volumetric and the gravimetric methods. And other miscellaneous methods include rate of reaction. This would actually use kinetic methods. And then if we are talking about heat or thermal characteristics, mainly we will use the thermal conductivity methods. Now, for the steps in quantitative analysis, we have here the flow chart. So the first one, when you try to conduct quantitative analysis, the first thing you need to do, of course, again, first or before the quantitative analysis, it should always have a qualitative analysis first. And again, as you try to establish the chemical species present or identify the chemical species present, then you may proceed with the quantitative analysis. And again, the first part of this quantitative analysis would be to select what method you should employ. So mainly, again, we have 4: gravimetric, volumetric, spectroscopic, or the electroanalytical methods. So when you choose a method, first and foremost, we may consider the accuracy that is required. But of course, if you consider a very high accuracy type of method, the drawback here would certainly be being time consuming and very much expensive. So if you have the money, if you have all the time you need, then you could opt for a method that could yield a very high accuracy. Another thing to consider would be the number of samples. So with that, again, time is of the essence. So if your sample preparation takes so much time, then maybe you could reduce the number of samples. But again, if you opt for a very high accuracy or precision, and for you time is not an issue, then you could have a lot of samples that you may like. Or if you have a method or instrument that could read a certain number of samples or a large number of samples, then you could opt for um, many samples to be gathered. Also, another thing to consider would be the complexity of the sample. Again, if you have time constraints, then you need to process your sample as fast as you can, or you need to employ methods that will process your sample as fast as you can. The next one, of course, is to acquire the sample. So acquiring the sample is what we know as the method of sampling. So in sampling, we should always consider that it would have the same composition as the bulk of material from which it was taken, meaning um, the sample should be a representative coming from the bulk of material that the sample was taken. And it should be a large and heterogeneous sample. And it could be a specimen from biological sources. And of course, when we have sampling, it is the most difficult step, since it is actually the most common source of errors. So if you do the sampling just right or correctly, then you could actually minimize or start to minimize the errors that you may obtain from this process alone and for the entire quantitative analysis. After acquiring the sample, then if it is necessary, you need to proceed with the processing of the sample. And the first step for sample preparation for solids would be the grinding, mixing, and homogenizing the sample. And if the analysis would be conducted later on, then you need to store your sample. Also, if your sample contains a lot of moisture, then you could do drying. And with that, you may that would be a result, or the drying is actually required if you have a gain or loss of H2O or moisture in your sample. That was for solids. Now let's proceed with liquids. So again, when you do sample preparation for liquids, tendency is that solvent may evaporate, and mostly you need to cover your sample so that it will prevent the contamination by, say, atmospheric gases present around your sample. So upon sampling or upon gathering your sample, you need to process the sample in such a way that you take good care of your sample by at most try to cover your sample and store it properly to avoid any evaporation of the solvent, and again to prevent any form of contamination. After processing your sample, or after preparation of your sample, we do replicate samples. So when you do quantitative analysis, it is very much imperative that we should have, or you should have, replicates of your sample. And with this, it actually helps you carry out your experiment in a precise manner. So at most you need to have, or the ideal one would be to have at least 3 replicates. And later on you will learn why we need to have at least 3 replicates of your sample. So again, when we do replicates, portions of a material of approximately the same size are carried through analytical procedure and at the same time and in the same way. So this is what it means to have replicates. And with that, it could result to a measure of the reliability of your results. The more replicates you have, the more reliable your results would be. And of course, you need to carry out an averaging or um, trying to get the mean values of those replicates. The third step would be the preparing of solutions. So what is required here is that you need to use a suitable solvent for each of your samples or for each form of your samples. And sometimes it will require chemical dissolution. If you have sediments or water samples, usually you try to heat it with acids or bases or other oxidizing or reducing agents. And also, upon preparing the solution, it is important that the solution or the resulting solution should have a property proportional to the concentration of the analyte in your sample. Next, after this one would be to eliminate interferences. So after processing your sample, you also need to eliminate possible interferences or the interference. When I say interference, these are the species that would most likely cause an error in an analysis by either enhancing or attenuating the quantity being measured. So it could increase or decrease or at most change the quantity being measured. So this interference will result to a bias or a very erroneous result in your chemical analysis. And to eliminate these interferences or interference, we most likely employ separation methods, and one of which, or the most commonly used method, would be the chromatography or any forms of chromatographic methods. Last one, no, second to the last or third to the last would be the calibrating and measuring of concentration. So we do a calibration of your sample. Later on, we will be discussing the important aspect of calibration. And here, this is an example of a calibration of your concentrations. So we do standard comparison, and with that, most methods actually employ certain calibration method because there's no direct way of measuring the amount of your sample, especially when we do spectroscopic or electroanalytical methods. So what we do here is mostly to do calibration or standardization procedures. After the standardization and calibration, then we will proceed with the calculation of the results. Of course, this mainly uses calculators and computers, and the basis for those calculations would be the raw experimental data, characteristics of the instrument, and the stoichiometry of the reaction. Later on in chapter 2, we will have a discussion on the balancing of chemical reactions and as well as the concept of concentrations and stoichiometry. So that after that particular topic, we could proceed with a more intensive study of quantitative analytical methods. And lastly would be to estimate the results or evaluate the results by estimating their reliability. So with this, you could measure the level of uncertainty of the results of your analysis. And For this to be measured or evaluated properly, we need to employ statistical analysis. And with that, we will have a thorough discussion of this part through chapter 4, 5, as well as in the early lessons for chapter 3. So that ends our topic for today. We will then proceed with lesson 1.2 later on. After this video or lecture video, we have an available assessment. Please check that one out before proceeding with lesson 1.2. Thank you. For the questions, you may address your questions via our Google chat or our Chem2 forum at our Google chat. Again, thank you.